Hello, my name is Rafał Wóycicki, and I would like to explain to you why it's so important to first have de-novo reference grade haplotype telomere-to-telomere reference sequences of the genotypes from the conditions that we are making our research on, and then use of structural reference genomics on this basis in human precision medicine or in plant and animal breeding. So first we need to know that same species genotypes from diverse environmental conditions have different genome structure. They differ in physical positions of DNA sequences on chromosomes. And for example, we have here same chromosome from two genotypes of the same plant species, one on the top from Southern Asia, where we have high light intensity, high temperature climate, and the chromosome positions from 2 million to 4 million with 5 different genomic regions marked with different colors, having 5 different genes. At the bottom, we have same genomic region from same chromosome, also same 5 genomic regions with 5 genes, but what we see here, it's not exactly the positions are not the same as the yellow genomic region in the Northern European climate genotype is rearranged between red and blue region. What it makes? It makes that the G1 is no more before G2, but between G4 and G5. And then what are the implications? So the most common SNP markers are generated with short-read whole genome sequencing, and their usefulness is confirmed both by checking segregation on mapping population and by their proximity to genes on the reference genomes. So making an example, we have breeding population from breeding company from Southern Asia, and this Southern Asia company is using actually the reference genome sequenced already, and this reference genome is sequenced from the genotype from Southern Asia. So they are first generating short WGS reads of the individuals of this breeding population, mapping this read, checking segregation on the mapping population, and then they are finding that marker A1 is linked to gene D2, marker MA2 to gene G3, MA3 to gene G4, and MA4 to gene G5. So now it's coming the very important example. Generation of genomic sequences from genotypes adapted to environmental conditions divergent from the one of breeding population results in erroneous linking of these sequences with genes and genotypes. How it's possible? So the breeding company from Northern Europe wants to find markers linked with threats. They have breeding population, they will make short-read WGS on the individuals, and they map these reads, but they map them to the genotype, to the reference genome from genotype of Southern Asia. They don't have the reference genome from Northern Europe. So what will they discover? They will not discover anything new, as the short read will map similarly. So the short read for marker MA1 will map in yellow region, and on the reference genome from Southern Asia, this region is close to green region. So marker MA1 is linked to gene G2, MA2 linked to gene G3, MA3 linked to gene G4, and MA4 linked to gene G5. But is it correct? As we know, it's not correct. It's just not correct because what they don't know is that European genotype has the translocation of the yellow region, and what it makes. The marker which was on fragment mapping to the yellow region actually is no more linked to gene G2 in the European genotype, because the yellow region is now between gene G4 and gene G5. So the marker on the fragment mapping on the yellow fragment is now linked to gene G5. and marker MA4 is no more linked to gene G5, but marker MA4 is linked to gene G1. So now, to be able to generate molecular markers working for the specific breeding population, it's necessary to map the WGS short read to the reference genome of the genotype adapted to the same environmental conditions as the breeding population. So first, it's very important to create the reference de-novo sequence of the genotype from the, on this example, Northern Europe climate, and then mark the short reads on this new specific reference. And then the company doing so would find marker E1 marked linked to the gene G3, IMI2 linked to gene G4, IMI3 linked to gene G1, marker European 4 linked to gene G5. And now it will be correct. As I mentioned at the beginning, the structural differences are not only in plants and animals, but it's also these are also visible in humans. So the publication from Gowan in last year, 2023, shows a large structural variations in between 36 Chinese subpopulations, which actually lead to differences in response to drugs, which is very important. So I hope that I explained to you the importance of reference genomics and structural variation. And if you would like to have more information, please feel free to contact me at appliedomics@appliedomics.com, and I will be happy to help you with both the de novo assemblies, comparative genomics, and the use of information gathered to plant-animal breeding or even human precision genomics. Thank you so much. Have a great weekend.